Hello and welcome. My name is Markus Langsdorf. I'm working as a product manager at biocrates. In this session, Professor Adamski and I would like to outline the challenges in human metabolomic studies and why it is so important to standardize quantitative metabolomics globally. In my presentation, I will explain why standardization matters under various aspects, also under pre-analytical considerations, and give an overview about the components of a standardized method. The best way to demonstrate the benefits of standardization are ring trials. I will talk about what it takes to make ring trials, what are the benefits, but also challenges. At the end, I will briefly introduce our standardized kits for metabolite quantification along with application examples. After my presentation, Professor Adamski will talk about the contribution of metabolomics in health and disease, and he will present the results of the global ring trial that has been carried out with one of our kit assays. I would like to start my talk with a prologue. Throughout history, humanity has developed various units of measure, many based on their own anatomy, such as fingers, hands, ells, or feet. Imagine if you would try to follow a set of instructions that requires you to measure 10 ells or a hand spread. If the application calls for precise and accurate tolerances, this is impossible. Your hands are a different size from the person who wrote the instructions. So if multiple people are working on the project, all with different hand sizes, the final result will be lost in translation or unusable. When we draw parallels to today's world, we find that just as different peoples had once used various arbitrary units of measurement, researchers in their own fields often depend on specialized in-house methods to measure metabolites and reproduce results. What we measure is of utmost importance, but actually how we measure it plays a fundamental role in shaping our ability to communicate, compare, and replicate findings across various labs and locations. So more than ever, there is a pressing need for a standard unit, a common language, so to speak, much like the metric system that emerged for physical measurements to enable precise communication among scientists and facilitate innovation and understanding. A common language would also help these two guys here who count the same set of bars but get to different results. So which result would you trust? Fostering trust in research findings can only be achieved with unified approaches and when everyone uses the same method. What else do we achieve with that? We achieve to minimize the variation and ensure consistent results across different batches, instrument platforms, and laboratories around the globe. Confidence. When different labs use the same method and protocol, their results are more believable. It facilitates a mutual understanding and enhances comparability in a globalized research community. We can share findings and learn from others where everyone speaks the same scientific language. And that finally enables faster progress when researchers build on shared standardized knowledge. Coming back to the picture, this shows why it is so important to have one clear way of doing things. When everyone uses the same method, we can trust the results more. Standardization also matters in pre-analytics. Especially there, small inconsistencies can have large effects and greatly influence the analytical results. So a standardized process is critical along the way from sample collection to analysis. Starting with sample collection. Consistency plays the major role here, in particular when doing comparative studies. Samples must be collected under identical conditions, for example, fasting, clotting time, or temperature, to reduce variability in results. Samples must be traceable and correctly labeled to allow for proper tracking and identification throughout the analytical process. It's also important to maintain their original state to ensure that results are comparable, even if analyzed at different times. We must ensure that the sample reaches the laboratory in its original or preserved state. At the end of the chain, proper sample storage is key to prevent degradation and to maintain the sample's composition. Here, I want to highlight the benefits of standardized methods under technical and other aspects. While lab internal methods, also called homebrew, offer the flexibility and innovation tailored to specific research needs, globally standardized methods will produce reproducible and consistent results when research is conducted in different labs under the same conditions. Other labs may have difficulty replicating the exact conditions and nuances of a homebrew method, leading to potential inconsistencies in results. The hardware components are based on optimized standard manufacturing processes to ensure batch-to-batch reproducibility. The validation on different mass spec types from different vendors is an integral part of the method development we do at biocrates. Cross-instrument comparability is an important criterion here. We follow guidelines for bioanalytical method validation from the European Medicines Agency when we validate our assays and kit technology. Findings based on standardized methods are more readily accepted by the wider scientific community. Homebrew methods, on the other hand, are often unproven, lack testing at scale, and results may not transfer well to other labs. So reproducibility concerns do exist. Sharing and comparing results becomes straightforward when researchers use universally recognized methods. With lab internal methods, communication can be complicated due to lack of familiarity, inconsistent parameters, or interlab variability. Another benefit is when people come and go in a routine lab workflow, the handover is easy as the method components and protocols are ready to use and also supported by the product supplier. Considering homebrew methods, manufacturing and method application often depend on individual competences. When people go, these skills often go too. Are actually the components of a standardized method as we develop and offer at biocrates. Basically, it's a set of procedures used for measuring analyte concentrations in biological samples to ensure comparability across instruments and labs, as already mentioned, or in other words, to minimize cross-lab and also batch-to-batch variability. The components are, starting from the instrument, specific validated methods with optimized parameters for best sensitivity and selectivity. SST samples consisting of a mixture of analytes covered by the assay for instrument performance check. Standard plate format for sample preparation. Calibration and internal standards for accurate quantification or analytical normalization to correct for potential losses during sample preparation or variability in instrument response. Quality controls, matrix-based materials of known concentrations for robust data normalization and longitudinal comparison of results. And the companion and guide for the whole process, a workflow manager and data processing software that guides through the whole workflow from sample registration through automated quantification and reporting. Now let's talk about ring trials. The validation of methods is improved through participation in inter-laboratory comparisons, such as ring trials or also proficiency testing for laboratory certification. Performing such trials is a requirement to comply with international quality standards, and they simply help make sure that science is reliable. So, what are the benefits and why do they matter? Ring trials are tests where different labs study the same thing to see if they get the same results. It also raises awareness of new technologies and methods and enables laboratories to trial a new method or test kit. They represent an external quality assurance and help us know if a method works well everywhere or just in certain places. So it provides assurance of a successful test transfer between laboratories. Is the method fit for purpose and suitable for science around the globe? Does it stand different instruments, lab conditions, and people? Ring trials are accepted by the community to prove the quality and reliability. If a method passes the test, many labs will want to use it. It's a unique opportunity to evaluate a method's robustness and universal applicability. When labs share what went right or wrong for them, we all learn and improve. Another aspect is that ring trials should be managed and evaluated by independent and neutral institutions or authorities. This, for example, was done with one of our kit methods and will be described by Professor Adamski after my presentation. This and the next slide, I would like to share some experience on what it takes to make a ring trial. There are a couple of to-dos in the whole planning, such as designing the experiment and defining the criteria. First of all, what is the objective of such a program? The key question is, does the assay deliver the same results in other labs and on other instruments compared to the own lab and own instruments? So the objective is to verify this, but also to verify the ease of use. Who should participate? It's beneficial to have diverse groups on board that represent various levels of expertise and, of course, different instrument types. Likewise, multiple sample types should be selected to evaluate method versatility, as well as spiked samples to assess accuracy, such as quality controls. Methods should be provided that have been validated according to accepted guidelines in analytical chemistry, such as from the European Medicines Agency, as mentioned. Also, the protocol must include detailed steps and clear descriptions. The metabolite quantification and reporting of the results must be standardized, what is automatically covered by our workflow management software in our case. The ring trial is completed by statistically analyzing and comparing the results and by evaluating the user feedback to identify technical issues and sources of error and to help optimize the protocol or workflow. However, a couple of challenges can come up, and there are some points that require careful attention. The participants should be encouraged to report problems and to ask questions, because not everyone is doing that directly. Some people are shy, some people are embarrassed, whatever. Regular meetings help here. Another experience is that not all participants follow the protocol stringently, so adequate training and support is required to be on the safe side. We must make sure that the samples are properly shipped and that, for example, dry ice is refilled in case the shipment gets stuck in customs. On the other hand, the participants must store them immediately at the recommended temperature, what is usually minus 80 degrees. An experience that certainly everyone here can share is that instrument problems or human mistakes can occur, what delays the trial or even impairs the results. Also, the best methods can create the variable results when the workflow or processing instructions have not been followed correctly. Especially different instrument conditions can lead to poor results. To a certain extent, this can be compensated by using internal standards or data normalization, but it's like a poorly tuned guitar. You can use a software algorithm to smooth the outcome, such as the data normalization in this case, but when the instrument is entirely off, it's getting hard. Timelines can also be challenging, as people often have to work on several projects in parallel. So sometimes rescheduling the experiment is necessary. As a last point, I would like to give you an overview about our standardized methods that are available as quality controlled and ready-to-use kits for quantitative metabolomics. The latest one that we launched in January this year is the Quant 500 XL kit. With more than 1000 metabolites, it has the largest combination of lipids and small molecules in one kit. With this, researchers will be equipped with a solution for a more comprehensive understanding of metabolite and lipid homeostasis in health and disease. It also contains hundreds of metabolites that are related to healthy aging and neurodegenerative diseases. With this kit, we organized an external beta test program before launch. 7 labs and 5 different instrument types participated here. The results are summarized in an application note that you can request from us. The smaller version is the Quant 500 kit with less lipids and with a panel related to nutrition and host microbiome interaction. About this kit and its ring trial results, Professor Adamski will talk in more detail in a minute. The P180 kit is on the market for already 13 years now and still in routine use in many labs. It has been established in hundreds of laboratories worldwide and is an excellent solution for high throughput basic clinical and epidemiological research. There was a ring trial with this kit in 2017, managed by the Imperial College London, with excellent reproducibility across 6 labs and 3 different instrument types. If you're looking for a specific solution to study microbiome and liver-related disorders, the bile acids kit is a good candidate. Also, with this kit, we performed a ring trial even across 12 labs with similar good performance. Thank you for your interest and attention. And now I'm looking forward to Professor Adamski's presentation.